All right, so we're going to be talking about the renal system today. And we're going to start out talking first about the different components of the renal system. The kidneys, of course, are part of this whole system, and they're found in the lumbar region of the abdominal pelvic cavity. And then sitting above the kidneys are the adrenal glands, and those adrenal glands are, as you already know, made up of the cortex and the medulla. They're secreting cortisol, aldosterone, epinephrine, norepinephrine. The kidneys are then attached to the ureters, and then those ureters are attached to the bladder, which is attached to the urethra. And the kidneys as well as the bladder are retroperitoneal. Now, you might think that retroperitoneal means that it sits behind the peritoneum, but that actually isn't the case. What that means is it sits outside of the peritoneum. So, for instance, the kidneys are sitting outside the peritoneum towards the back of the body or the dorsal area, whereas the bladder is sitting outside of the peritoneal membrane and it is anterior in the body. So it doesn't mean just outside of, it doesn't mean in back of, it does mean outside of. So we're going to talk about filtration today. We're going to talk about reabsorption, mention some secretion. Then we're going to go into excretion and, of course, micturition, which is urinating. Now, when the kidneys are filtering blood, your blood goes through the kidneys. The entire blood amount in your body goes through the kidneys about once every 5 minutes, and you're filtering about 180 liters of blood a day. Now, 99% of everything that you filter goes back into the bloodstream, and that's something that we call reabsorption. So if it's filtered out of the blood and then put back into the blood, reabsorption is occurring. And so that means that about 1.5 to 1.8 liters a day are excreted. So a person makes about a 2-liter bottle of urine per day. Now, the kidneys are made up of a cortex, which is the outer portion, and this cortex is where the actual filtration and reabsorption is taking place. And then we have the medulla. Now, the medulla is only made up of these long tubes, which are called the collecting duct, and they're kind of like the sewer system that's bringing the urine from the cortex, where the nephron is located, and all of this filtration reabsorption is occurring, and then bringing all of that urine that's produced into the renal pelvis and then into the ureter, the bladder, and eventually the toilet. Now, in order to do this filtration, we have to obviously have blood supply, and that blood supply is coming via the renal artery, which is bringing in essentially the dirty blood into the renal cortex. And then the filtration occurs, and the cleaner blood, and I say cleaner because it's not going to be perfectly clean of waste products, but the cleaner blood is going to come out through the veins and then leave by the renal vein. Now, the kidneys, their function is to filter out what we would call metabolic waste products. So whatever waste products made in the metabolic chemistry of the body, so whatever metabolism is being produced, like for instance lactic acid and uric acid, the kidney's job is to filter that waste product out. And again, most of the waste products that we make are going to be acidic, so it's very important to get those acidic products filtered out, because those acidic products lowering the pH too much could then, of course, cause denaturing of proteins. All right, so here is the sequence of arteries, and we go from the aorta, renal artery, all the way down to the afferent arteriole, glomerular capillaries, efferent arteriole, peritubular capillaries, and then via the veins to the renal vein to the inferior vena cava. Now, the only ones that you need to be aware of that we're really going to talk about and focus on are the afferent arteriole, the glomerular capillaries, the efferent arteriole, and then the peritubular capillaries. The rest of them, I'm sure you had to learn those in anatomy. We're not really going to worry too much about them. All right, so now, what we would call the functional unit, or where all the work is being done, is in an area that's called the nephron. And in kind of yellow color here, beige, this is the nephron. It's made up of what we call the renal tubules. These tubules are very small. They're microscopic, so instead of calling it a tube, we refer to it as a tubule. And then these renal tubules also interact with, and this is in red and blue and kind of purplish color here, these are the various blood vessels that are necessary for that filtration reabsorption process to occur. So what you're looking at here is you're looking at two different types of nephrons. This first nephron right here is referred to as a cortical nephron. Now notice these blood vessels right here. Okay, these are called the arcuate blood vessels, and I'm going to go back to the picture we just went through and show you these are the arcuate blood vessels. So the cortical nephrons are sitting in the cortex here of the kidneys. And so that's what we have here. These are the cortical nephrons. There's a little bit in the area which is the medulla, but most of it is in the renal cortex. This second nephron, this is called the juxtamedullary nephron, and part of this nephron is sitting in the renal cortex, part of this nephron is sitting in the medulla. All right, so the first thing we're going to talk about as far as the nephron is concerned is the process of filtration, which again is moving fluid, moving substances from the blood into the lumen of the nephron. All right, so the next portion that we're going to talk about that deals with filtration is the glomerulus. And the glomerulus is made up of essentially two components. The first is the renal capsule, or the glomerular capsule, also known as Bowman's capsule. And Bowman's capsule is made up of two parts to it. There is a visceral layer and there is a parietal layer. The visceral layer looks like each one of these little cells looks like an octopus here. These are what they call podocytes, and they are attached to capillaries. The parietal layer is a layer that goes all the way around these capillaries, and kind of looks, if you look over here, it sort of looks like a basketball that goes around the capillaries. And this makes up all of Bowman's capsule. Now, inside Bowman's capsule are what are referred to as the glomerular capillaries. And the glomerular capillaries are called that because glomerulus means a ball of yarn, and somebody looked at it and said, you know, they all look confused in there, so they look like a ball of yarn. And so we have here the afferent arteriole that's leading into the glomerular capillaries, and then we have the efferent arteriole that's leading out of the glomerular capillaries. And we need to take some time and talk about both the glomerular capillaries and then the arterioles. So in this picture here, what you see is you see the glomerular capillaries. And these capillaries are what we call fenestrated. They're a special type of capillary for a couple of reasons. One is because they have little tiny holes in the walls of the capillary. The second really important thing about these glomerular capillaries is the blood pressure. So if you notice, the blood pressure in these glomerular capillaries is about 55 millimeters of mercury, which is nearly double what the blood pressure is in other capillaries in the body. So very, very high blood pressure, which allows for water filtration to occur. So you can see here how I've drawn this. The water is actually being pushed out of these capillaries due to the high blood pressure. Matter of fact, this blood pressure is so high that if nothing stopped the force of the blood pushing the water out, then your fluid in your blood would be drained in no time, and a person would die from hypovolemic shock. But we have what is referred to as the osmotic, or sometimes also called the oncotic pressure. And this is due to the protein albumin. Now, albumin acts like a sponge, and it pulls water back into the capillaries and prevents us from getting this hypovolemic shock and dying. So the albumin draws the water, the osmotic pressure draws the water back into these fenestrated capillaries. So you do have some filtration of water occurring, but most of the water is coming back into the capillaries. Now, you also have, due to the water being pushed out, something called solvent drag. And that means that a lot of the ions and smaller particles are able to move out of these capillaries, just diffuse out and be drug out basically by the water that is being pushed out by the high blood pressure, what we'd also call hydrostatic pressure. Now, there is a problem with this because you don't want to lose that albumin. And fortunately, albumin is a pretty big molecule, and those fenestrations in the capillaries are pretty small. So for the most part, albumin isn't going to be lost through the fenestrations. However, there are times where there is a possibility of albumin loss. And for instance, if somebody is sick, let's say maybe they have a bacterial infection in the kidneys, those fenestrations can get larger, and there is, possibility of protein ending up in the urine. So, when we do the little dipstick thing, that dipstick is really testing, one of the things it's testing for is albumin in the urine. And if that albumin leaves, then there is a possibility of excess amounts of filtration of water out of the system, and so that would not be good. So in order to protect the body and to make sure that we don't lose too many proteins, or any proteins I should say, from the glomerular capillaries, there are a couple of different things that happen. First of all, number one, the fenestrations are very small. Second thing is, all the way surrounding these capillaries, all the way around the outside, is what's referred to as the basement membrane. And the basement membrane is made up of a bunch of negative charges. Now, proteins overall are, for the most part, negatively charged. So if albumin were to be able to, let's say, slip out of these fenestrations, then the whole idea is that this basement membrane would allow the albumin to be repelled, or force the albumin to be repelled back in because of the negative charges. So the negative charge of the basement membrane, the negative charge of the albumin, albumin would be repelled back in. Now, if that's not good enough, because obviously, remember, having this protein inside the blood is very important in not allowing it to escape. We also have the podocytes, and these are those little octopus looking like cells that surround the outside of each one of the capillaries, the glomerular capillaries. And these podocytes have these little teeny tiny slits here. These are called filtration slits, and they're very, very small. And so what happens is, if that albumin somehow is able to get through the fenestrations and then somehow penetrate through this basement membrane and the negative charges, then the next thing it would have to do is get through those filtration slits. And they're very small, and typically they're not able to get through. So if there's protein in the urine, obviously that is very meaningful, because there's a lot of things that have to break down in order for the albumin to make its way into the urine. So here's the glomerular capillary. This is showing us the outside of the capillary, and then this capillary is attached to another tubing system where the fluid that's going to be filtered, the metabolic waste products filtered out of the capillaries, is going to end up in this tube. So we'll talk about that. It's called the proximal convoluted tubule as we go along. So in the glomerular capsule, okay, this would be considered the parietal layer. And then we have the glomerular capillaries coming into this capsule. So you have what's called the afferent arteriole, bringing blood into the glomerular capillaries, and then filtration occurs. And the fluid that is being pushed out by that hydrostatic pressure, we're going to refer to this as filtrate. We're not going to talk about this yet as urine, but this is the filtrate, and we'll change it up. We'll make it a little bit different before it finally becomes urine. And then the blood is going to leave through what's called the efferent arteriole. Now, there's another thing that's really important about all of this, and I'm going to go back to this picture back here just for a second and talk about the afferent and efferent arterioles. But before I do, let me just mention, this is the podocytes, and you can see here just a little bit the filtration slits, and I'll show you another picture of that in a second. But these afferent and efferent arterioles are very interesting because they're a different setup than any place else in the body. In the body, usually we have arterioles that go into capillaries, and then from the capillaries they go into venules. Here you have an arteriole that goes to a capillary that goes to arteriole, and then it goes to another capillary system, which we'll talk about, called the peritubular capillaries. And from the peritubular capillaries, it then goes into the venules. So the question is, why do we have this weird setup? Why do we have arteriole, capillary, arteriole? And the answer is there's a difference between arterioles and venules in the amount of smooth muscle. And because of this smooth muscle, we can control the blood pressure in these glomerular capillaries. So if the afferent arteriole were to vasoconstrict, and the sympathetic nervous system can release epinephrine and norepinephrine and cause these afferent arterioles to vasoconstrict. The question is, what would happen if the blood pressure went up in the afferent arteriole? What would happen to the pressure in the glomerular capillaries? Well, think about this constriction here, because it's kind of like the hose at your house. Let's say that you were to turn on your hose and you wanted to get something off your driveway. And so you're going to use the hose to squirt down the driveway. And so the question is, how are you going to get the pressure of your hose to go up if you've already turned it up all the way that you possibly can at the spigot? Well, typically how we do it is we put our thumb on the end of that hose. And if you put your thumb on the end of the hose, you've just made the diameter of the hose much smaller. And by making the diameter smaller, the pressure of the water coming out is going to be higher. And then you'll be able to clean off your driveway. Now, the same thing happens with the afferent arteriole. If we constrict that down, it's like putting your thumb over the hose. The blood that's coming out of the afferent arteriole into the glomerular capillaries is going to have a much higher pressure. The higher the blood pressure in the glomerular capillaries, the greater the filtration. So if we go back to this picture here, remember, blood pressure is already at 55 millimeters of mercury, but if we make that pressure go up, we're going to force the water and whatever other chemicals are small enough to get through all these fenestrations and the basement membrane and the filtration slits. Whatever other chemicals can be pushed through, we're going to even be able to push more. So we're going to increase the rate of filtration, or we'll increase what's called glomerular filtration rate, or the GFR. Now, on the other hand, what if we vasoconstricted the efferent arteriole? Well, we're going to have the same kind of concept, except this time the fluid is going to be coming out very quickly from the efferent arteriole. So there's a much higher pressure which pushes the fluid out, which now means that the pressure is going to drop in the glomerular capillaries. With that drop in pressure, there's going to be a decrease in the glomerular filtration rate. If the GFR goes up, then you're going to produce more urine. If the glomerular filtration rate goes down, you're going to produce less. So think about this for a second. What if you have a patient who has hypovolemia, low blood pressure? Do you want the afferent arterioles to vasoconstrict, or do you want the efferent arterioles to vasoconstrict? Take a second, try to figure that one out as we go on. All right, so we've talked about changes in the glomerular capillaries. And so again, this is our glomerulus with our afferent and efferent arteriole and filtration actually occurring. And this is just another picture of the glomerular capillaries. They're fenestrations. And then this is one of the podocytes from the visceral layer of the Bowman's capsule. And then these little teeny slits here, these are the filtration slits. So again, if anything gets out of here, then it's got to be able to get through this very small fenestrations, through the negatively charged basement membrane, and finally through the filtration slits of these podocytes. Not an easy task to do. Now, if something is able to filter out of the glomerular capillaries, the next place that we're going to see this filtrate enter is into a region called the proximal because it's close to the glomerular capillaries, and this is a convoluted or very twisted tubule. So this is the proximal convoluted tubule, and these tubules have a whole bunch of microvilli. Now, these microvilli are always on cells when absorption of some kind of chemical is going to occur. In the case of the proximal convoluted tubule, it's going to help us to reabsorb substances. So this is what we call transepithelial transport, which just means that it's diffusing across the cell membrane. And reabsorption can occur actively, it can also occur passively, which we'll talk about both of those. So with tubular reabsorption, we're looking at here the wall of the proximal convoluted tubule, and there are certain chemicals, certain things that have been filtered that are going to be reabsorbed across the wall of the proximal convoluted tubule, and this time back into the peritubular capillaries. We're going to use sodium potassium ATPase pumps to do this for active transport. Then we're also going to just simply use concentration gradients, osmotic gradients for passive transport. So this is talking about sodium reabsorption through the proximal convoluted tubule, and here's the lumen of the tubule. Now, when filtration occurs, we know that water is being pushed out of the glomerular capillaries, and with that water, certain things like sodium, glucose, amino acids, certain vitamins, bicarbonate, and waste products, of course, are all being filtered out as well. Now, you don't want to lose a lot of these products in the urine, and the reason that they're filtered out is because these are very small particles and they're able to get through all of the various layers in order to make it into the tubing system. And unless we do something about this, these particles are going to end up in the urine. And so we're going to talk about how do we get these good things reabsorbed back into the bloodstream. And so in the tubing system, inside the proximal convoluted tubule lumen, you have a very high concentration of sodium. However, in the cell itself, the concentration of sodium is much lower. So by simple diffusion, we have sodium moving into the proximal convoluted tubule cells on what we call the basolateral surface. So this would be the basal surface. This is the bottom. Here's our microvilli. This is the basal surface, and really this picture isn't drawn very well. It should be more towards the side here. This is the lateral region. On the basolateral surface, we have sodium potassium ATPase pumps. When sodium diffuses from the lumen of the proximal convoluted tubule into the cells, the sodium is then going to be actively transported out of the cells and then into the peritubular capillaries. So on the apical surface, just simple concentration gradient, high to low. And then on the basolateral surface, we have the sodium potassium ATPase pumps. Now, glucose is one of the really important chemicals that we need to reabsorb back into the system. So glucose also is going to move down its concentration gradient with sodium and diffuse into these cells. And then glucose is going to basically piggyback with sodium and move across active transport with sodium back into the peritubular capillaries. So apical surface, this is co-transport with sodium. Basolateral surface, the glucose is using the movement of sodium to get back into the peritubular capillaries. This is a slide that's out of place, so I'm going to come back to this in just a second. And here's another slide out of place, so we'll skip that for now. This is again just showing you a picture of the peritubular capillaries. And so this is just my drawing. These are the cells that make up the peritubular capillaries. Here's the proximal convoluted tubule, and these are the microvilli, and then on the outside you have the peritubular capillaries. Okay. The filtrate, again, this has water, sodium, glucose, amino acids, waste products. These have all come out of the glomerular capillaries. And on our basolateral surface, we have our sodium, potassium, ATPase pumps. And sodium is going to diffuse high concentration to low into the cells of the proximal convoluted tubule. And following that would be water by osmosis. It's following the sodium. And then sodium is going to be actively transported, pulling the water back in. Now, if we just look at sodium and water before we go back and look at glucose and amino acids, by the time the filtrate moves through the proximal convoluted tubule, 75% of all the sodium and 75% of all the water that was originally filtered out of the blood is going to be reabsorbed back into the bloodstream. The glucose also is going to follow sodium and diffuse in. The amino acids are going to follow sodium and diffuse into the proximal convoluted tubule cells. And then just like glucose that can piggyback across on sodium, the amino acids will do the same thing, and they piggyback across with the sodium. Waste products will diffuse into these cells, but they will just diffuse back out again. They do not have the ability to use this active transport pump with sodium and then to piggyback with sodium back into the bloodstream. So these waste products, even though they'll diffuse into the cell, high concentration to low, then they'll diffuse back out high concentration to low. Now, by the time the filtrate leaves the proximal convoluted tubule, if everything is healthy, we'll have absorbed 100% of all of the glucose that was originally filtered and 100% of all of the amino acids that was originally filtered, so that you should not have any glucose in the urine whatsoever in a healthy person. So that means if we're going to do a dipstick test and we do a dipstick and look at glucose levels in the urine should be 0. Unfortunately, that's not always the case. Sometimes we do dipstick tests and there is some glucose in the urine. So the question is, what in the world is going on? And that goes back to the slide that we passed up, and instead of going back to it, I'm just talking to you about it now. Glucose, when it piggybacks across with sodium, there's only so much glucose that can get across per minute. The sodium, potassium, ATPase pumps are pumping at a very particular speed, and glucose can only piggyback at a very particular speed. So let's pretend that we have somebody who has diabetes, for instance. We know that in diabetes, there's not enough insulin. We can't move glucose into the cell, which means we're going to have a lot more glucose floating around in the bloodstream. Now, if that's the case, if there's more glucose in the blood, that also leads to an increase in the amount of glucose that's filtered out of the blood and ends up in this filtrate here. So you have more glucose in the filtrate than normal, but you have a sodium-potassium pump that can only move glucose across at a certain rate. So that means that this extra glucose is going to end up wasting in the urine. Now, glucose is a particle, and one of the things we talked about a long time ago was that water follows particles. This is an osmosis thing. So one of the things we see is if a person has excess amounts of glucose in the urine, they're also going to have a lot of water wasting. And so in diabetes, we see that people have a lot of urine output, or what's called polyuria. And then because they also have a lot of urine output, they also will end up having low blood pressure. And we talked about before with blood pressure that one of the things that happens with the low blood pressure is you get this unquenchable thirst. And that's exactly what people with diabetes have. Active diabetes causes unquenchable thirst because all this water is being transported with the glucose into the urine. And so they have what's called polydipsia now. And of course, the other one is polyphagia, which simply means that the person is constantly hungry because they don't get enough glucose into their cells. So it's the 3 P's of diabetes: polydipsia, polyuria, polyphagia. Okay, so this next part we're going to talk about is the loop of Henle. So we've gone over the glomerular capsule, and then we've talked about the proximal convoluted tubule, and this tubule leads into this loop of Henle. Now, we have 2 portions to the loop of Henle. We have what's called the descending limb of the loop of Henle, and the filtrate is going to move through this descending limb and then up into the ascending limb of the loop of Henle. Now, the peritubular capillaries, if we go back here, notice in purple, these are winding all around, okay, the nephron. By the way, if they're winding around the loop of Henle, they're also referred to as the vasa recta. And so I have the peritubular capillaries here off to the side so that you can see those. Now, the descending limb of the loop of Henle, I want you to know this is only permeable to water. The only thing that can escape through this limb is water. Whereas on the ascending limb of the loop of Henle, we have sodium, potassium, ATPase pumps. And the only thing that this limb is permeable to is sodium. So only sodium is going to be actively transported out of the ascending limb. Only water is able to leave by osmosis from the descending limb. Very important. So first of all, we'll talk about the ascending limb. The sodium pumps are constantly actively transporting the sodium out of the ascending limb. Now, keep in mind, we're talking about this in 3D, so this is this tubing system, and all around this tube in 3D is sodium. Now, the sodium is constantly being transported out, which also means that eventually around the descending limb, we're going to see sodium buildup. So we get this really high concentration of sodium in what we call the interstitial space, and that sodium eventually is going to simply diffuse high concentration to low into the the peritubular capillaries, or the vasa recta. Now, because the sodium is diffusing, well, first of all, because the sodium's in high concentration around the descending limb, water follows sodium by osmosis, first into the interstitial fluid around this limb, and then back into the vasa recta, or the peritubular capillaries. So again, you have sodium being actively transported out of the ascending limb, building up in the interstitial space, and then eventually just simply diffusing into the peritubular capillaries. Water from the descending limb follows just simply by osmosis. So that now, okay, when we left the proximal convoluted tubule, we had reabsorbed 75% of all of the sodium, 75% of all of the water, and 100% of the glucose, 100% of the amino acids, and 0 of the waste products that were originally filtered. In the loop of Henle, the only thing we're looking at here is sodium and water. And by the time the filtrate leaves the loop of Henle, we've reabsorbed about 15% more of the original filtered amounts. So now we've reabsorbed about 90%. But let's think back for a second. I told you in the very beginning of this video that we filter our blood once every 5 minutes, and altogether we're filtering out about 180 liters a day. So if we were to stop here with the filtrate we've made and just have that as our urine, a person would be urinating out about 18 liters of urine per day. Now, instead, we're going to need to cut that down. And so there's 2 more parts now to our nephron. There is a distal convoluted tubule and a collecting duct. And these are going to take care of that last 10% of sodium and the last 10% of water. So, again, let's look at this. This is our glomerular capsule with our afferent efferent arteriole. Here's our proximal convoluted tubule. Here's our descending limb of the loop of Henle that just has the permeability for water. and the ascending limb of the loop of Henle, which is actively transporting sodium. And then here's our distal convoluted tubule, which then is going to connect to the collecting duct. Okay, so we're going to talk about the distal convoluted tubule. We're going to talk about the collecting duct in a second. I just want to show you here, as the filtrate moves down, notice these numbers show us how much more concentrated the particulate matter inside becomes because we're losing water. But as the filtrate moves up through the ascending limb, as we're losing sodium because it's being actively transported out, you can see that the concentrations start to go down. All right, so talking about the distal convoluted tubule, this is the distal convoluted tubule here. It attaches to the collecting duct. And I just wanted to point out the afferent and the efferent arteriole. The afferent arteriole comes close and touches the distal convoluted tubule, so you can see it better here. This is our afferent arteriole, our glomerular capillaries, our efferent arteriole. And then these are very important. These are the juxtaglomerular cells. And we have within this region baroreceptors, and these baroreceptors are going to detect changes in blood pressure in the afferent arteriole. And the juxtaglomerular cells are then going to secrete an enzyme called renin, which mixes with angiotensinogen to convert it to angiotensin 1. And then angiotensin converting enzyme, or ACE, converts that to angiotensin 2. And then angiotensin 2 attaches to the adrenal cortex, which then makes aldosterone. And then we have sodium reabsorption. Now, if you remember all of that from our blood pressure essay, that's what we're going to be talking about now. So blood pressure control has to do with decreased perception of blood pressure in the afferent arteriole and the release of renin. Angiotensinogen is converted to angiotensin 1. Angiotensin 1 is converted to angiotensin 2. And then we'll have aldosterone and antidiuretic hormone secretion occurring. So this is representing our distal convoluted tubule. It is attached to the collecting duct. And then surrounding the distal convoluted tubule and by the collecting duct are the peritubular capillaries. In the distal convoluted tubule, like we've seen before, we have sodium-potassium ATPase pumps that can actively transport sodium back into the bloodstream. Now, these pumps are a little bit different, though. These pumps can change speed. So the other pumps we've seen so far in the proximal convoluted tubule and in the loop of Henle, they're just moving at a particular pace. Not the pumps in the distal convoluted tubule. These pumps can move faster or slower so that we can reabsorb more or less of that final 10% that was originally filtered. So the question is, what's controlling this reabsorption of sodium? That would be aldosterone. Aldosterone, when it's secreted, can change the speed of these active transport pumps so that we can reabsorb all 10% or much less than that full 10%. Also, antidiuretic hormone is involved with the collecting duct. There are valves on the collecting duct that can open if antidiuretic hormone is present. If this hormone is present, the valves open, and then water, by osmosis, is going to follow the sodium back into the bloodstream. If water follows the sodium in, again, depending on how much sodium is in that bloodstream, then we could potentially reabsorb all last of that 10% of water. So we could reabsorb 100% of the water that was filtered, so you have 0 urine output, or you reabsorb, on a normal basis, you reabsorb 9%, so you have about a 1% urine output, which again is about 1.5 to 2 liters of urine per day. Excretion is where we're getting rid of the waste products. That's what this term means. And then micturition is where urination actually occurs. And one of the things I want to talk to you about is that there are stretch receptors in the bladder. And these stretch receptors, when the bladder fills with urine, they're activated and send sensory information via the sensory neuron to the lumbar region of the spinal cord. Now, the lumbar region of the spinal cord then sends information via motor neurons to the internal and external sphincters. Now, these sphincters are a good thing to have some control over because you don't want to have to be wearing a diaper all day, but babies do. They have to wear a diaper, and the question is why if we have a motor neuron there? The answer is because the information to the central nervous system is not completely hooked up until about 18 months of age, which means that the stretch receptors will send information to the spinal cord, but then the spinal cord cannot send that information up to the higher centers of the brain because the reflex just isn't there. And so what happens is the information just goes straight to the motor neuron and tells the motor neuron, hey, you need to relax, and then the baby wets their diaper. After about 18 months, there is information going up to higher brain centers, and then the question is, hey, do you want to relax those sphincters or not? And it might not be an appropriate time to relax them, so the brain says, no, just wait. Now, once another 50 milliliters of urine enters into that bladder and it stretches again, then we're going to question the brain, are you sure you want to wait? Now, sometimes, unfortunately, incontinence can occur. One of the reasons for incontinence is pregnancy. The way that the uterus is tilted, it's tilted forward right on top of the bladder and can push, when the baby gets bigger, can push down onto that bladder and cause the sphincters to open. Or we could have what's called stress-induced incontinence, which simply means that due to stress, the sphincters open, and so if a person laughs or runs or whatever, they may end up urinating themselves. All right, so that is all for today for our renal system, and good luck to you.